Hi, everybody. Welcome to our new season of VanBug. I'm very excited that we're we are able to keep this going and with our new slate of speakers this semester. So we'll start off by this is how our schedule is going to be. Offset it by 5 minutes. So a bit of announcements from me, and then some introductory student talk by Shreya, and then the main featured speaker by Tao Huan, and then it's going to be open for Q&A. Just a few announcements. First of all, we've changed our mailing lists. So we did migrate your, if you've been subscribed to the old mailing list, we've migrated that to the new mailing list server. But if for some reason you're not receiving any of the announcement emails, go to our website, and then there's a mailing list tab, and just follow the instructions, which is just basically sending an email to this SFU address, and that would automatically subscribe you to the mailing list. And you can unsubscribe by just sending an unsubscribe subject to the same email. So for this season, we plan to hold the whole season, 2021-2022, until the spring over Zoom. And we have a full slate of main and student speakers booked for this season. You can check it all out on the schedule page on our website. And we do have plans for December events. Probably it's going to be some kind of lightning talk, something that we've done 2 years ago that was really exciting. And we are thinking about bringing back, but more details will be announced soon. And of course, VanBug would not happen without the generous support of our sponsors, including the Graduate Studies in Bioinformatics program that a lot of you are part of, and Genome BC, and Ecoscope at UBC, the bioinformatics.ca website, or the Canadian Bioinformatics workshops, and the UBC spinoff Accelera. And we are this year also excited to have a partnership with Langara College with their new bioinformatics program that they are starting. And another new sponsor is Stem Cell Technologies that we have this year. So thanks for all of the sponsors, and thank you all for attending VanBug. And as a reminder, VanBug is affiliated with ISCB and our two sister bug organizations, Montreal and Toronto, Monbug and Torbug. So you can check their websites. They also have most of the seasons they're running it online this year as well. So you can check out those as well. So with that, I'm going to quickly introduce our speakers. First, we have Shreya Bendre. She's a graduate student at Dr. Bernard and Lynn Lab, at Drs. Bernard and Lynn at the BC CRC. And the topic of the talk is going to be targeting fibroblast activation in fibroblast activation protein alpha for cancer imaging and radiotherapy. And sorry for this typo in the title. And our main speaker is going to be Dr. Tao Huan. Dr. Tao Huan is an assistant professor at the Department of Chemistry at UBC. He received his PhD from the University of Alberta in analytical chemistry with a focus on chemical isotope labeling based metabolomics. And after that, he did his postdoc at the Scripps Research Institute in California. And now, since 2018, he's been an assistant professor at UBC in the Department of Chemistry, working on synergetic development of analytical and bioinformatic methods for mass spectrometry-based metabolomics. And many of you might know him through his affiliation with either the GSAT program or the Genome Sciences Center, or the grad bioinformatics program that many of you are of. So with that, I would like to thank our speakers for showing up and for presenting to us. And just as a general rule for our speakers, anybody, if you have a question, type it in the chat. Speakers, feel free to answer it, address it throughout the talk. But if you also want to leave it to the end, and if we have time, then I'll read out the questions. And if somebody wants to raise their hand to ask a question by themselves, then me or any of the other moderators would be able to handle that.
Screen, right?
Yes, now it's full screen. Yes.
Okay, great. I think it's really good to meet all the bioinformaticians and the students doing bioinformatics in UBC and SFU. So today I'm gonna talk a little bit about the work that we've done in our lab to address the big data challenges in mass spectrometry-based metabolomics. And for people who are not familiar with metabolomics, it's actually very similar to genomics, transcriptomics, and proteomics, in a way that it is a study the study of the entire set of small molecules in the human body. So, metabolomics together with other omics can be used to understand health and disease, and they are very good hypothesis generation tools for us to understand the biological questions. And among different analytical platforms to study metabolomics, the most commonly used one is the liquid chromatography mass spectrometry system. In the LCMS system, the LC system is used to separate the metabolites based on their different interactions with the stationary phase, the column stationary phase. Then these molecules will be converted into charged ions through this ionization process. Usually we use electrospray ionization. It is a soft ionization technique that will not break down molecules, so you will have the molecules in, it will be intact, it will just add a charge on the top. So then once you have charged molecules and get into the mass spec, and then you can just do the measurement. And in the LCMS analysis, we will have the LC chromatogram. This is what typically you will see in the experiment. The x-axis, the time, or we call the retention time. And for each metabolite, it means how long it stays in the LC column before it gets detected by the mass spec. And the y-axis is the abundance or mass spec signal. So if you have your mouse, you will move over and click one of these data points, you will have this MS spectra displayed here. And as you can see, at each time point, we can collect a large amount of metabolic information. Each value here represents one metabolic feature, represented the M over Z value of the metabolic feature. So usually in one MS spectra, we can get over 100 features per spectra. And then in one LCMS analysis, we usually collect 10,000 MS spectra. So you think about it's 10,000 spectra, and within each spectra we have more than 100 features. So it is actually a large amount of data that is waiting there for us to process them. And as you can imagine, this is not something that you can process manually. So we have to rely on software and bioinformatic tools. And then we think about the LCMS platform in the entire metabolomics workflow. We will first prepare the samples. We extract the metabolites from the samples. Then we perform LCMS analysis. Once we generated a gigantic amount of data, we need to use software to process them. Then we need to choose the appropriate statistical tool to extract statistically significant features for downstream identification as well as biological interpretation. I mean, LCMS is a very sensitive platform, and it allows us to collect a large amount of data. But the large amount of data also become a big data challenge in metabolomics. Especially these are the few difficulties here that right now limit its applications. So first, because you have so many data, we cannot do the experimental optimization as we've done in the past. If you only focus on, say, compound A, right, you can easily check a different experimental conditions and see which condition give you the best extraction efficiency. Now we are talking about thousands of tens of thousands of metabolic features, and the optimization become challenging. And another thing is for all these metabolic features, some of them are known, some of them are unknown. When we use bioinformatic tool to extract them, it is very difficult to make it 100% extraction efficiency. You will always have some features left in the raw data, and these low abundant features could be very important for your biological interpretation. But unfortunately, they can be extracted, and you can't do interpretation for them. And then how we determine quantitative values, and how we clean up spectral contaminations, and how we confirm a metabolic identity. So there are many challenges there because of the big data issue. Back in the older days when people do analytical chemistry, because they already know what they are looking for, they have a targeted list of metabolites or targeted list of chemicals. So it's usually very easy for them to say, okay, see, this metabolite should be in the sample. Now we couldn't find it. Then let's go back to the experimental protocol to optimize it. But now if we are talking about thousands of features, that's become impossible, and which leads to the big data challenges that are waiting for us to solve them. So our lab basically mainly focused on 3 different directions. One is how can we extract more metabolic features from raw LCMS data, and how can we improve the quantitative accuracy and the precision, and finally how can we annotate these unknown metabolites. And in my following slides, because this is a talk to the bioinformatic scientists, so I basically will pick some of the work that's more related to bioinformatics. So first of all is the metabolic feature extraction. So on the top here, this is a typical chromatographic peak shape for a metabolic feature. And this feature shape, it follows, usually it's a Gaussian shape. Because of that, people have developed the strategies to extract them by recognizing its Gaussian peak shape. So like in this work, LCMS, which is one of the first automated peak picking algorithm, so they use a second derivative Gaussian shape to fit the peak. So any peak that belongs to a metabolic feature, they should be able to be fit by the second derivative Gaussian shape. And from there, we can tell, okay, here we go, we have a metabolic feature here. But the problem is many metabolites, they have very low abundance in the biological sample. Therefore, their chromatographic peak shape might not be good. As such, they cannot be fit by the Gaussian shape. So this leads to a problem is that all the conventional data processing software, they recognize the Gaussian peak shape. Yet we have lots of metabolic features, they don't have a good chromatographic peak shape, and they just cannot be extracted by these conventional software. And this is an analog, is like iceberg, right? And I mean, those are the good ones, they can be picked up by software, but the large amount of features, they don't have a good peak shape, they cannot be picked by conventional peak picking software. So, and that leads to us to think about how can we extract these low confidence features, because sometimes, these low confidence features, they contain important biological meanings, right? It could that lead to some bigger biological discovery. So this leads us to develop a called integrated feature extraction strategy. We first use those conventional peak picking algorithm to fit these Gaussian peak shapes to extract level 1, level 2, and those are high quality metabolic features. On the other side, we also use the unique MS2 spectra from the raw LCMS data. And these MS2 spectra, they don't have the, they have a unique precursor ion at those level 1 and level 2 features. And then we extract those features out as level 3 metabolic features. And by combining level 1, 2, and the level 3 features, we are able to achieve a better metabolomic coverage. And this is done through bioinformatics, not about analytical method development. But I think this is very critical because using this approach, we'll be able to extract about ranging from 30% to 100% more metabolic features from the raw data. And furthermore, we also incorporated, peak picking and MS2 recognition. And also we allow users to input a target list, a list of metabolites that they think that they should be found in the sample. And using this approach, combine, we call it joint data processing, combining peak picking, MS2 recognition, and a target list. We are able to actually extract even a longer list of metabolic feature. And also, this is our package. We basically developed a R package, and also we have a customizable spectral library, so users can do automated metabolic annotation based on their study. So then, people in the field, in the metabolomics field, or people who are familiar with the mass spectrometry analysis might have the question is, you are able to extract those low abundant features, but how about their fidelity? How about their quality? Are they real metabolic features, or are they just a background noise? And to address this thing, usually what we do is through manual checking. And for us as a human, after you've been trained in metabolomics for a few years, you can immediately tell, okay, these chromatography peaks, they represent a real metabolic feature because they have a very good peak shape, but those ones have a bad chromatographic peak shapes, and they are for sure not real metabolites, and they are more likely to be background noise. Okay, this is easily done, but now we are talking about thousands of metabolic features, and it is unrealistic to manually go through each one of them, right? So that leads us to develop a machine learning algorithm or convolutional neural network. We train a convolutional neural network model to automatically recognize metabolic features with good chromatographic peak shapes from bad chromatographic peak shapes. And using this model, we develop it is on the back end, it's a Python program. And we also made a C# interface. So users, all they need to do is just click a few buttons. And this tool is also available on Microsoft Apple Store. And we believe this is the first metabolomics tool that's available on Microsoft Apple Store. So to prepare, to do this analysis, all you need to do is prepare the raw LCMS data, and you input the raw data into our program called EVA, which is short for evaluation. And on the other side, you can use these data processing tools to process and generate feature table. Then EVA will automatically retrieve their chromatographic peak shape from raw data for these features in the feature table. Then the algorithm will automatically recognize whether this is a good shape or bad shape, and then finally give you a percentage, as well as each the recognition classification for each spectra. And the beauty here is we trained the model is very robust because we trained it with over 25,000 spectra. And thanks to the undergrad students who work in our lab over the summer, they manually go through each one of them, and we trained 25,000 spectra. And there are many ways you can use EVA. One thing is to check the quality of data. Another thing is to filter those false positive features, like the one shown here, right? The false positive feature. Using EVA, you can exclude it out from downstream data processing. And then this also allows you to automate the parameter optimization. Say, for example, you input a set of parameters for peak picking, then you realize it has a very high percentage of bad peak shapes, then you can tune the parameters. And EVA allows you to do this all automatically, right? You save you a lot of time. And also using this tool can allow us to develop a better peak picking algorithm. You can have a better algorithm and using EVA to evaluate the spectral quality. Okay, from there, I want to move on to talk about data quality. So fold change is commonly used in biology, right? We measure the concentration of a metabolite or protein in one control sample, then in a disease sample, we calculate fold change. Then we say, okay, they have twofold increase in the disease, which means this protein or this metabolite could be important to the disease. And in mass spec analysis, we also use the same idea. We basically measure the metabolite using mass spec, and we recorded the mass spec intensity in the control in the disease. Then we calculate the fold change. Then we say, okay, the fold change is too big. Then it might be an interesting metabolite. But then this is actually something people haven't recognized that, although people doing mass spec for quite a long time. So here's the example I show you. So for a linear, so we are talking about a linear ionization range, right? You have a calibration curve here, and for a particular metabolite, sample 1 present a metabolite at this level, and sample 2 present metabolite this level. And you can easily tell for these two samples, their metabolic fold change is 2. In another way, sample 2, the metabolite in sample 2 is about 2-fold of the metabolite in sample A, metabolite in sample 1, right? But if you think about their mass spec signal intensity, I mean, this is a linear, okay? Nothing to do with nonlinear. It's just a linear, in the linear range. The measured signal fold change is only 1.2, right? So, I mean, usually when we do fold change, we use a threshold of 1.5. And if you use mass spec signal, you will say, okay, fold change to 1.2 means that this metabolite is not significant. But in reality, it's a real fold change to 2.0. So why that is the case? And how can we solve this problem? So then the way we first did is we collect 20 over 20 metabolites, and we prepared to have a fold change. So basically, we prepared 2 samples. One sample has a concentration of 5 nanomole, another sample 2.5 nanomole. Right. So then we measure their mass spec intensity. So in principle, their metabolic concentration difference is twofold. Then we have their mass spec signals. Then we can use their signal to calculate their signal ratio or signal fold change. And we find that most of them that are actually outside of this 2.0 fold change. And only a small amount of them are within the range. So then the question is why we have so if the fold change is smaller than the real fold change, we call it the fold change compression. If the fold change is larger than the real fold change, we call it fold change compression. So why we have compression and inflation? So why we would have this compression and inflation? So actually, we did a further investigation. It's actually related to the intercept. So when you have the intercept larger than 0, you will have a compressed ratio or compressed fold change. When the intercept is equal to 0, it's unbiased. No matter what slope you have, it's not biased. But if the intercept is smaller than 0, your measured intensity ratio will be larger than the real concentration ratio, or we call it inflated ratio or inflated fold change, meaning that the measured mass spec fold change will be higher than your real concentration ratio. And we actually did a more systematic study. We have the urine sample. 100% is our original urine sample. We diluted all the way to 10%. Then we have 10 different concentrations. Then we compare the mass signal of each of these diluted sample against the original one, and we will be able to get the concentration ratio. For example, if you have 100% versus 10%, the concentration ratio is a 10, right? Because the concentration fold change is 10-fold, and it's located here. And if you compare 100% to 50%, the fold change is a 2, right? So you have a 2 here. And we have the red dashed line that represents the expected signal ratio or expected fold change. And then from the metabolomics experiment, we have thousands of metabolic features, and they have their signal intensities. So we can actually, using these signal intensities, to calculate their MS signal ratio. And we plot their distribution in the violin plot. As we can see, most of those signal ratios are actually deviated or biased, right? They are deviated from the real expected signal ratio. So this is actually a big thing, right? Because in many cases, when you do mass spec analysis, you just need the mass spec signal to calculate fold change. But our study showed that the fold change is biased, and probably you shouldn't want to use that. So how, and also another study we did is now we have 10 different samples. We basically compare, we just randomly pick a pair, and we compare their expected fold change and versus their versus the mass spec signal fold change for thousands of metabolic features. We find that actually quite a lot of them, as shown, the ratio bias larger than 10%. We have quite a lot of percentage of those metabolic features. They have a biased signal ratios or fold change. Okay, then how can we solve this problem? To address this question, so we basically use quality control samples. The quality control samples are prepared by taking a small aliquot of each individual sample and put them together. And then for the quality control samples, we prepared different concentrations or different volume. Then we can use this quality control sample to build a calibration curve for each metabolic feature. Our underlying assumption is that if a metabolite exists in a real biological sample, it should also exist in a QC sample, because the QC sample is a pool of all these individual samples. And then because we have the calibration curve for each metabolite, then what we did is for a metabolite, detected in the individual sample, its intensities can be converted to QC injection volume using this calibration curve. So therefore, we can convert all the intensities to the injection volume. Then we calculate the fold change using the converted injection volume or QC loading amount. By that way, we will be able to solve this fold change compression or fold change inflation issue, because the QC loading amount should be proportional to the metabolic concentration with an intercept of 0, right? And using that, we can solve the problem. Okay, so further to that, and I want to talk about a general concept. People, if you took a second year undergrad course, like we're teaching here at UBC, you know that the total variance of analytical experiments is always related to the variance of analytical variance plus biological variance. Analytical variance is basically, sample preparation, right? Sometimes you add a little bit more, just some variation from analytical aspect, and also biological aspect. People to person, have different blood pressure, have different oxygen concentration in the blood, right? So these are the blood biological variation, and this concept has been held true for hundreds of years. People basically believe that's the case, but we claim that in omics research, there's one more thing we should consider. Because in omics research, we also need to consider computational variation. And the question is where the computational variation come from. So it is actually from the, because we need to get a quantitative value, right? And the quantitative value usually is a peak area or peak height. Because now in omics research, we do not measure these values manually anymore. We basically do a peak area integration or peak height calculation using software. But as you know, software sometimes can make mistakes. If they integrate the peak area or calculate peak height in a not appropriate way, then you will have this computational variation. And because you have a computational variation, sometimes statistically significant features can become a statistically non-significant. So for us, how can we correct this, right? To have a smaller computational variation. And one example we provided here is that we also, again, get some chemical standards. We did LCMS analysis, and we manually calculate the peak height and integrate peak area, as shown in these blue dots. And also, we automatically determine the peak height and peak area using a software, MS Dial, which is very commonly used data processing software. So, so these are the plotted. So we have analytical replicas. I think we have 5 replicas for each. So you can calculate RSD for each quantitative measurement, and then for their RSD we can calculate make a box plot. And it's showing the box plot. The automated approach always generates a larger RSD than the manual approach, and this difference is contributed to computational variation, right? Because the same sample, right? It's the same sample, so it's not analytical variation. There's no biological variation. Then it's a quantitative computational variation. And we also find an interesting phenomenon is actually when we compare the RSD values of a peak height-based quantification and peak area-based quantification, we find that for most features, the peak height-based quantification is more accurate than peak area-based quantification when we use MS Dial to do the process. So how can we solve this problem? And ultimately, I want to say is to develop a better peak picking algorithm so to accurately determine where the peak starts, where the peak ends. That will be the ultimate solution. But this is outside the scope of this paper. I think there's a long way to go to achieve that. So for this paper, what we did is to get some easy solution, which is, again, using QC sample. So the way we did it is for all the biological and the QC samples, after LCMS analysis, we calculate the intensity tables by peak area and by peak height. So we have two tables. Then for using the quality control samples, we can actually compare the RSD, the relative standard deviation of the peak height and the peak area for each metabolic feature, right, using the quality control sample, because QC sample is basically replicated injection. And we believe that if the QC sample has larger RSDs in the peak area than peak height, then we probably should use the peak height, because peak height has smaller computational variation. And alternatively, if the peak area has better performance than peak height for the quality control samples, then we should also use the peak area for the real biological samples for quantitative assessment. Okay, so from there we get a mixed table, mixed by peak area and peak height. H represent height, A represent area. It's a mixed table. And at this stage, of course, you cannot directly do quantitative comparison, right? Because they have different magnitudes. So then we also do a calibration and conversion, like the one we talked about in full change correction. We just have a different QC loading amount for both area and height. Then we do this, we fit this information and we calculate their loading amount. Now they all been converted into loading amount. This allows us to make a fair comparison of the samples. And I mean, this is not the best solution, but at least this is a doable solution. So one result we show that, so this study is some patient sample before chemotherapy and after chemotherapy. And then we find that, so as shown here, we have we have some 263, they are uncalibrated features, means that they don't need to do calibration, they're already good enough. And then we have some they are being calibrated, and some are significant, some are not. And let's jump into here. So we find the before versus after correction. We find the 4 metabolites before the correction. The p-value is large, and it is usually considered statistically non-significant. But after we correct its computational variation, we get much smaller p-value, and which makes it we basically pick it into this significant list for downstream biological interpretation. Okay. Then we also thought about, this might be something you already learned in your first year graduate course, is that the statistical performance in mass spec analysis sometimes is called diminished by the nonlinear ESR response. Nonlinear ESR response is a common phenomenon in mass spec-based analysis, means that if you keep loading more and more and more higher concentration of the sample, the metabolite response will not be linear all the time. It will be curved. Sometimes it could be curved towards this direction. Other cases will be curved towards the other direction. But basically, it's nonlinear. Okay, and because of nonlinear, we assume if the metabolic concentration is linearly, it's a Gaussian distribution, right? Symmetrical. You have group 1 and group 2. Okay, then you measure the mass spec intensity. But because of the nonlinear response, your normally distributed concentration will become either left skewed or right skewed metabolic signals or mass spec signal. And this skewed metabolic signal will causing problem for downstream statistical analysis. And this is something that people already know, but there is no comprehensive study of how this nonlinear ESF response really affected the skewness of mass spec signal. So the way we did it here is we use the data simulation and the Monte Carlo simulation. Basically, we simulated the two groups of normally distributed data. Then we input an ESI response that we can calculate using regression method from our experiment. Then we can convert this concentration data to intensity data. And we did a Monte Carlo simulation. Basically, we simulate the process 1,000 times to improve their robustness. Then because as see in these two groups, right, we can calculate their p-value. In this case, you can see these two groups are really far away from each other, and its p is definitely smaller than 0.01. But it might not be the case after we convert them to the intensity table. So the way we did it is we have the intensity table. We can recalculate the t-test, right? We can calculate the times of p smaller than 0.01 out of this 1,000 simulation. and that gives us a true positive rate. And actually, we repeated this experiment 15 times. So in total, we have a 15 times 1,000. So this is actually very interesting because the true positive rate is a common way people use to estimate the statistical power of the power of the statistical method. And we also tested different sample size. As we can see, in all these cases, the true positive rate is basically of the concentration is always higher than the true positive rate of the intensity. The effective size, if you are not familiar, it's basically how these two groups are separate from each other. The larger the effective size means these two groups separate far away from each other. If it's a 0, means these two groups overlap with each other. And as you can see, when you have a larger sample size, it will help, right? When you have a larger sample size, when you have a effect sample size, when you have larger effect size, the true positive of the concentration-based, the intensity-based t-test will be the same after a certain level. That totally makes sense because you have a bigger significant difference. But when you have the sample size is small and the effect size is not that big, the intensity-based t-test will have a significantly dropped compared to the concentration-based. And in this case, we used a nonlinear ESF response that combines the convex and concave shape. Okay, now we notice that this is a problem and how can we solve it? That's related to our development of a PowerU workflow. And in the PowerU workflow, so we first calculate their response. And then from the response, we first do a normality test to see if the mass spec signals, they follow if it has high normality. If it's high normality means it's not skewed, then you can just do normal t-test. Otherwise, if they are not normally distributed or if the data is skewed, then unfortunately you have to do a U-test because the U-test is a non-parametric test. So it will have a lower statistical power. But because the data is abnormal, there's nothing we can do about it. So we have to use a U-test. So basically, in all, the PowerU workflow allows us to choose between t-test and U-test depending on the data normality. And we find out actually using PowerU workflow, we'll be able to detect more significantly change in metabolic features. And there's one example is shown here. So some of you, familiar with the statistic, they might ask, why don't we do log transformation, right? Log transformation can solve the skewed data. But actually, that is only half of the story. This is what the log transformation does. If the data is skewed, right, then you do log transformation, it will make it bring back to normal. However, in metabolomics, because of the different types of nonlinear ESF response, you have a different type of skewness. For left skewed data, log transformation is perfect, no problem at all. But for right skewed data, the log transformation will actually bring the skewness to even worse situation. Okay, so the question is, how can we develop a better transformation method that to make the data looks more normal? And then we talked to our colleagues in the East Coast, professor in statistics. They said, oh, why don't you try Box-Cox transformation? And this is what the Box-Cox transformation looks like. So the transformed data basically is whatever the value you have, you have the power of lambda -1 divided by lambda. The lambda is a tunable parameter. And this is the how the Box-Cox transformation looks like. And when lambda equal to 0, actually you can do the calculation. The Box-Cox transformation based on Len transformation, or in our words, log transformation based on E, right? So basically, Box-Cox transformation you can consider as a more universal transformation method, and then log transformation is just a special case for Box-Cox transformation. Okay, now we have this thing here. We developed adaptive Box-Cox transformation. Why we call it adaptive? Because different metabolic features, they have a different skewness direction. So we need to find the best lambda, or we need to optimize the lambda based on the data distribution. So this is how we do. So we have actually several other steps on the top of that, but I think it's not relevant to, not quite important here. So the most important is how we optimize lambda. The thing is for a metabolic feature table, right? You have multiple groups, and each group you have a multiple, signal intensities. And from there, you can do the normality test to get its initial p-value. Then we actually have the lambda value. We iterate from +3 to -3 by a factor of 0.01. So you basically use the lambda value 3, put it into the ABC transformation. We call the ABC transformation to recalculate the normality. If it gets a better, then we basically calculate the normality for all these values and then pick the one that have the best normality, and that becomes your optimized lambda value for the Box-Cox transformation. And we use that to generate the transform the table for downstream t-test. Okay, so the benefit of, so here is just one example of the performance. We actually have a given distribution. We have normal distribution, positively skewed, including exponential and the chi-square distribution, and negative skewed, include not normal and the beta distribution. Then we do the data sampling, and we use the Monte Carlo simulation. And then once we have the data, we transform by log square root, and this log transformation and square root transformation is commonly used in metabolomics, and then of our ABC transformation. Then we tested the normality again after the transformation, and repeat this 10,000 times. Then we calculate the data normality rate. You know, the percentage of out of these 10,000 experiments, how many of them can actually generate a normal distribution. So for normal distribution, no problem. You don't even need to do the transformation. For a negative skewed, so negative skewed is actually the left skewed example. So this is people call negative skewed. So negative skewed case, when you all these transformation did a great job, but the positive transformation, as you can see, ABC transformation outperformed others. And in reality, ABC transformation, because it's more robust, it can tune the lambda value based on your data distribution. So no matter what conditions you have, the ABC transformations always outperform other data transformations. And we also did a application, you know, now the fancy COVID sample, we received some COVID sample, we calculate the normality distribution, you know, before transformation and after transformation. So before ABC transformation, after ABC transformation, and we can see no matter it's a normality or skewness, so after ABC transformation, it always gets much better. And then this is for all the metabolic features, right? They optimize the lambda value, and this is distribution of the lambda value. As we can see, the lambda value is usually between -1.5 to 1.5, suggesting that our initial setting of -3 to +3 should be enough to cover everything. And actually, we can see after ABC transformation, we can always detect more statistically significant features. And two examples showing here, those metabolites are actually related to the lung inflammation, further indicating the power of ABC transformation in preparing better metabolomics data. Okay, so now let's switch the gear a little bit because we always talk about metabolomics. A key step here is to do statistical analysis, right? And but this is not something that the people like in biology, because in biology they already know what metabolite they are interested in, right? They want to just target this. And how can we do this biology-driven metabolomics? So the idea here is how can we perform on-target metabolomics on a class of metabolites, you know, both known and unknown, that are related to a given biological question, right? And the example we showed here is steroids. And the steroids can be either chemically synthesized or produced by plants, you know, bacteria, fungi, or, you know, or human bodies. And it's very essential signaling molecules, and the dysregulation of steroids related to disease, and also synthetic steroid, right? Those people, you know, doing sports, you don't want them to take synthetic steroids because then the result will have problem. So how can we do that? So that leads us to do a machine learning model that can automatically recognize the steroid features from LCMS spectra, regardless of the steroids being reported in literature or not. The idea behind is all the steroid molecules, they share similar chemical structures as presented here, right? They all have the 4 benzene ring and also some side modifications. So because of that, their MS spectra, if you do the fragmentation, their MS2 spectra also look similar. Because of that, we can actually train a model, a CNN model again. And in this case, we have an in-house spectral steroid spectra, and also we collect some public available MS2 spectra. And also we have because when you train a model, you need both positive data and negative data. So we have steroid MS2, non-steroid MS2, and a bunch of steps to improve the data quality, which is also the key feature of that paper. But because of time limit, I'm not going to talk in details. But anyway, basically, we generate CNN model that is very can be used in the real application, that you input your raw LCMS data, right? You have all the MS2 spectra, then the CNN model can automatically make a decision, whether this is a steroid like a spectral, this is not steroid like spectrum. So from there, you can generate a feature, and then from there you can do a biology-driven metabolomics if you're only interested in the steroid molecule. And also we did a cross-validation. Our results show that this model is very robust, and it should be okay for biological application. And the one thing we did here is we take a urine sample, we did our LCMS analysis, then we can actually detect using our steroid extractor bioinformatics software. We'll be able to find 135 steroid spectra, and among the 100 of them can be annotated, to have a structure like a steroid-like structure, and the 69 annotated the ones actually being reported in the literature. So here's the important thing, right? So the male versus female, if you do PCA plot using all the metabolic features detected in LCMS analysis, there's no separation. Basically, the male and the female metabolites, overall, they have a very few difference. But if you extract their steroid features and redo the PCA plot, the principal component analysis, you will find that the steroid features of male actually can be clearly separated from the steroid features of the female. So this is basically using machine learning algorithm to do the automated biology-driven metabolomics. So finally, I want to talk about, because we do metabolomics, but we're also interested in link our metabolomics result with the genomic and proteomics data. So one way we do, so this is the feature table we always get, right, from our metabolomics result, and usually we will just send it to our biological collaborators. And in most cases, they are not interested in this table because this table only has a value, but there's no biological meaning. So the thing is, how can we get the biological meaning from this metabolomics table, right, for biological interpretation? So that leads to the metabolomics guided system biology paper work that we did back a few years ago and published in Nature Methods. So we first incorporate the biological pathway prediction and also multi-omic data integration. Unlike the conventional approach where people need to do statistical analysis, identify the metabolite, then do biological interpretation, this is a time-consuming step because the identification part really takes time. What we did is we want to incorporate some algorithm to directly get their biological pathway information from the metabolic features prior to metabolite identification, so that biological researchers, they can maybe pick up some pathways they are interested to further perform metabolite identification. And this is achieved through this work, biological pathway prediction that published by Professor Suzhao Li at Emory University. So basically, it used Fisher's exact test to predict the MS2 structure from this feature table. And from there, because now we can directly predict the metabolic pathways as well as metabolic network from features table, and you can immediately tell how these metabolite information they are related to each other. And from there, you can get this cloud plot where the x-axis is the overlap metabolic features, y-axis is the significance of these different pathways. And from there, we think it's also important to do multi-omic data integration. Unlike people in genomic and proteomics, they first predict the pathway using genomic data, then do multi-omic integration. What we did here is we first predict the pathway using metabolomic data, then we overlay proteomic and transcriptomic data for multi-omic integration. And in particular, we have those genomic and proteomic data, they are already publicly available, which make it easier for us to do this job. And in this column cast example, we can actually download the data from the TCGA and then do the analysis, and then generate the multi-omic system biology result. And also, we are actually making some new development in the lab to further incorporate genomic proteomics for system biology studies. Okay, so with that, I would like to conclude my talk here. So I hope through my presentation here, I can convince you that the big data challenges in metabolomics is addressable. Basically, we need the knowledge not only analytical chemistry but also bioinformatics, computer science, as well as biology. And we can take some of those existing tools from genomics, proteomics, because they've been developed much longer than us. And finally, I think it's important to collaborate with people, bioinformaticians, to further advance this work. So finally, I would like to thank all the funding agency and all my team members who make this work possible. And finally, thank you for your attention. I'm looking for your questions.
Thank you so much, Tao, for this great talk. And I guess while waiting for questions to trickle in, I have a couple of questions. Like at the beginning, you were introducing metabolomics and like talking about the similarities to them to other omics that we have, and I'm wondering. Using the techniques that you're like the techniques that you're dealing with, like with mass spectrometry, for example, where are we at like being able to link what you're seeing there, especially for like metabolic targets, metabolites that are novel, or you don't know what you're looking for, but you're able to just see their spectra, to proteomics or even transcriptomics. Where are we in the ability to link these different players?
Right, that's a very good question. There are actually two major steps that still need to be addressed. So first is because actually I didn't talk about that actually, because in metabolomics we detect a large number of unknown metabolic features, and those features can be contributed from some biotransformation reactions, the microbial biotransformation, or other, the food intake, those some exogenous metabolites that are not being found in the human body yet. And to confirm and identify these metabolites, big challenging, and the people have been developing tools to do this job. So that's the first thing, we need to confirm these metabolites. And the second thing is the multi-omic integration. At the current stage, most of the multi-omic integration is rely on the pathway. But as we know, many of the metabolites, they are not really involved in the endogenous pathway. And how can we, do the integration? For example, people can do network analysis based on correlation, and that is one possibility. And I believe there is a very big room there for us to develop a new bioinformatic tool to address this multi-omic integration challenge.
And when you say confirm, like what does that entail? Like, is it is it confirming by like trying to like replicate by using the same techniques, or is it confirming using some orthogonal methodologies?
Right. So that's also a unique challenge in metabolomics, because for genomics and proteomics, for example, in gene, right? Once you know the sequence, you can easily map to the library. They will tell you what the gene is. For protein, if you know the peptide sequence, you can also do the database search, because all the protein, they are made of 20 amino acids, and the protein can be predicted from the DNA sequence. But metabolite totally different. Metabolites have a diverse chemical properties, and we have the known chemicals, millions of known chemicals. Also, there are more unknown chemicals there. So that's one difficulty. You cannot predict the chemical structure from protein. That's one difficulty. And another difficult part is in mass spec analysis, we basically measure only the M over Z value, or we only tell the molecular weight information. And that molecular weight, because for same M over Z value, for same molecular weight, it can have a multiple isomers, right? It can be structural isomers, steroid isomers, and So we have to confirm, say for example, out of these 20 isomers, which one is the real compound, right? And on the separate story is that because when we do the measurement, there's always a variation, right? So when we measure the molecular weight at 200, it could be 200 plus minus 0.005. I mean, even it's a small difference, but that difference can lead to multiple candidates, and we want to confirm them. And that part is also a very challenging part.
I see. Yeah. I mean, I complain about transcriptomics, but this sounds like a much like a bigger nightmare, or basically like a darker nightmare.
Totally. Think about transcriptomics and genomics, right? Now we already have commercialized the product, the 23andMe, for example, and also we have a precision medicine. People have been using genomic information. It's already start to get commercialized, start to get into public use, but metabolomics is still at the stage where we're still trying to develop a method. There are still something people are not quite sure what's the best way to do it. So we are about 5 to 10 years lagged behind the genomics and the proteomics.
One of the things that you've talked about is when you're looking at these like spectra, and then like you have like the signals, and then there's this like the good signal and the bad signal, and like trying to like you have like this machine learning approach to classify them. But my question is like, what is happening in these bad signals? Is these just like random noise, or is there data in there that like we're only detecting what we are familiar with, like even like the annotation in the end, like annotating it as a good or bad is based on what we're expecting to see in there. But do you have any ideas about like what could there be like in these bad signals that are actually like representative of something actually happening in the molecules?
That's right. So that is also another challenge, because mass spec is a very sensitive instrument. So during your sample preparation, if any contaminant you get into your system, they will show up as a signal in the mass spec. And those signals, it's very hard to be, it's hard to tell whether it's from the system or from the real sample. And people have been developing bioinformatic tools to tell the real features from those contaminant features. So that is one thing. And another thing is lots of these unknown metabolites, because people don't know what it is, and it showed up in the mass spectra. And sometimes people will say, oh, this might be a contaminant, but actually it is not. And right now, the way to tell whether this is good or not is still mainly based on the manual checking and based on the expertise of the persons who's looking at the data. But I think gradually, people are trying to develop machine learning tools to automate the process. Yeah.
Cool. I guess we have our first audience question. So it's from Giuliano Cruz. Sorry if I'm mispronouncing your name. Thanks for the great talk. I wonder how the normality tests prior to downstream analysis may impact your overall error rates. For example, your FDR. On the same line, can't you perform the same picking strategy based on the gamma distribution, which handles the skewness issues rather than normal, in order to avoid the need for transformations? And sorry, just as a note, if somebody would like to speak up, just raise your hand and I can let you speak. So sorry, back to you, Tom.
Okay, yeah, so there are two questions. The first one is how the normality test affect the overall error rate. I'm not quite sure, because normality test is down to the metabolic feature table, right? It only shows you whether it has a high normality or whether normality is good or not. I'm not sure how it's related to the error rate. So basically, if you have a high normality, then you are likely to have a less error, like less false discovery. Oh, you're talking about false discovery, right? Okay, so that will be a separate topic, because false discovery relate to the multiple testing scenario, right? Because we do thousands of features, then you have a false discovery rate. And that will be a separate topic from the the normality test we performed. We perform normality test to make sure that when we do the t-test, we have a normally distributed data so that we can maximize the t-test performance. Yeah. Then for the second question, can you perform peak picking? Are you talking about the metabolic peak picking? I mean, there are many different ways of doing the peak picking, but the problem of if the peak picking is performed based on peak quality, there will always be a chance that the low quality peaks will not be picked up. And that is separate from the skewness. The skewness is about the quantitative result. So the quantitative result can be skewed because of nonlinear ESI response. Let me see. Yeah, so these are two. The peak picking is one thing about how you extract features from the data. And then the skewness is more about the quantitative part. You know, the quantitative measurement can be skewed because of the nonlinear ESI response. Okay.
We have another question from Eric Navarro. So thanks for the talk. When integrating multi-omics data, are all of the significant results weighted equally? For example, would you keep only the pathways that have significant results in all transcriptomic, proteomic, and metabolic data, or would you have a preference for selecting some pathways that have significant results in one or two of the omics data, even if it's not significant in the others?
Yeah, that's also a very good question, actually, because of time limit, I didn't get a chance to talk more about the LCMS system biology platform. So it's actually a web-based platform. You can log into the LCMS system, you upload metabolomics data, you finish the metabolic feature extraction, right? Then you get a feature table. Then you can upload the genomic proteomics data, and then there's a lot of different visualization tools you can choose. And it actually outputs everything, and including the significant pathways as well as the non-significant pathways. And the users, it's actually kind of have a interactive interface. So users can click each pathway and see what the metabolites are get involved, and they can click the metabolite to see the box plot to see the statistical results. So it's very interactive. It actually have everything there.
Cool. So we are approaching the end, but I do have one last question. And if somebody has a question, please submit it right now, and then we'll wrap up today's event. And my question was on one of the topics that you've discussed is using quality control samples, especially when calibrating for the log change, log fold change. And my question is like about the as a procedure, like how expensive is this to add this extra like control here, and how many quality control samples do you typically need? Are 2 enough, or do you need like in the example you showed us like up to 10?
Right, so there are actually 2 types of quality controls. I mean, so first, of course, you take a small aliquot from each sample and prepare the quality control. Then there's 2 directions to go. So first, you need to run the quality control sample every few injections of a real biological sample. And this is to use the quality control sample to check the instrument performance because over the time the instrument signal or drifted, then you can use the quality control sample to correct the signal drift. So that's in that case, it's basically same quality control sample, but did the injection after every few sample. Usually we do every 10 sample injection, then we do 1 QC injection. Then to prepare, for example, the full change correction, the PowerU Workflow and others, then the users need to take the quality control sample and do serial dilution. And usually we need to dilute at least 5 different concentration values so that it can cover a wider concentration range. And that QC, like a serial diluted QC sample, can be run at the beginning of your LCMS sequence. So you can run a serial diluted QC sample, then you start to run with biological samples. And then after every 10 biological sample, you run 1 more QC sample. So basically, there are 2 different ways to use the QC samples. Yeah,
I see. Cool. Well, thank you so much, Tao Huan, for your great talk and like opening up our season. And also thank you for our introductory speaker. Shreya, and that was really great. And hope to see you all next month. And remember, if you didn't subscribe to the mailing list, the new mailing list, please do on our website. And thank you everybody for attending.
Bye.